Okay, thank you. It's a real pleasure to be here. So I want to frame what I'm going to talk about as sort of a war between HIV, a virus, and our immune system. And I think that it's clear that HIV is winning, except in the cases where we have drugs to intervene. So I'm going to start with reminding you of the life cycle of HIV. And, okay, yeah. So HIV is a retrovirus, which means that it reverse transcribes its RNA into DNA. So the way this all starts is there's a virus out here, which is going to infect this cell, and it infects the cell through the interaction between its spike trimer. I'll be calling this the envelope or the env, or I'll call it a variety of things. That binds to the host receptor, which is CD4. And the fact that, well, I also want to define that within the spike trimer, I'll be talking about GP120, that's one of the subunits, and also GP41. So GP120 is the part that binds to CD4. Okay, so this part here binds to the host receptor CD4, and that restricts HIV to cells that express the CD4 marker, which is, of course, a very important molecule in immunology. It's on a subset of T cells. So part of the reason HIV is a problem is it tends to wipe out CD4 positive T cells, and then your immune system is completely dysregulated. Okay, so it binds to CD4, and that triggers a conformational change in the spike trimer that allows it then to interact with its co-receptor, which is called some of them, CCR5 or CXCR4, these are chemokine receptors. Okay, so I'll be talking about the interaction between GP120 and the spike trimer as a whole with CD4, and then also the conformational change that occurs to allow it to bind to co-receptor. But so we've only gotten to the part where it binds to the outside of the cell. What happens after it binds to CD4 and CCR5 is that fusion takes place between the viral membrane, this is an enveloped virus, and the host cell membrane, and that allows the genetic material of HIV to get into the cell, where it's then reverse transcribed by its own enzyme reverse transcriptase into double-stranded DNA that's integrated into the host genome, and then when you start making virus, the genes are transcribed and translated, and then you get more viruses out. So fortunately for us, there are a number of ways to inhibit HIV in someone who's infected. So as a triumph of structural biology, really, there's been small molecule drugs designed that inhibit the viral reverse transcriptase, that inhibit the viral integrase, the viral protease, and then there are some fusion inhibitors as well. But people have to take these as a combination, which is called highly active antiretroviral therapy, or HAART. They have to take usually at least 3 different drugs, because the reverse transcriptase of the virus is so error-prone that a huge number of mutations arise within every cycle of viral replication. And so if you have just one drug, the virus can immediately mutate so it's resistant to it. So if you give 3 in combination, the virus can't mutate fast enough to come up with 3 different mutations to evade all 3 at once. So that's the common course that's given in this country and in developed countries around the world. The problem is, if you look around the entire planet, only a little over 1/3 of infected people are being treated with these available drugs as problems due to accessibility and stigma and so on. So it's really not working that well. It could work, but it doesn't work that well, and there's also side effects. Really what we want is a vaccine. This would be the ideal way to protect against acquiring HIV. So the way the vaccine would work, if it would work, would be you'd inject something into the host, and then their own immune system would make antibodies. So that's the way most vaccines work. So this is depicting, here's the virus right here. The only target against which you could raise antibodies that would neutralize a virus would be the envelope trimer that I introduced in the last slide, and so here's the antibodies out here. So if you could raise these antibodies, then the antibodies would bind to the virus and then either block the attachment in the first place or they'd block the fusion or something. But in other words, they would block the virus from infecting the cell, and these are called neutralizing antibodies. The problem is, anyone who's infected with HIV makes neutralizing antibodies. That's not a problem, but most of them are strain-specific. And there's a huge number of strains in even a single infected person. So that's what I'm trying to depict here. Within a single infected person, there's this, what would be called a viral swarm that's created by the error-prone reverse transcriptase. So this is illustrated here by different colors. And so antibodies will be raised that will neutralize these viruses. But for example, this blue antibody would neutralize only the blue viruses or maybe the purple ones, but it wouldn't neutralize, let's say, the red ones. So that's a big problem because HIV continues to mutate against these antibodies here. And so it's like a race where the virus mutates faster than the antibodies can keep up with. So in the past 10 or more years, a number of laboratories have started to characterize antibodies that they get from the blood of HIV-infected donors. And so I've taken away the viruses now, and so now you look at these antibodies. This is done through single-cell cloning and then sequencing of the genes. If you make the encoded antibodies, then you can test them in vitro and ask how broadly neutralizing they are. And so, unfortunately, most of the antibodies that people get from patients are strain-specific. But there are rare antibodies from rare patients, and these are broadly neutralizing. And by that, I mean that they neutralize, let's say, more than 50% of the strains that you might test in the laboratory. So to give you an idea here of how many strains there are, I suppose no one really knows, but there are more strains of HIV in a single infected person than there are strains of influenza virus in the entire world. So this is a problem with trying to make a vaccine, obviously, and it's a problem with isolating these broadly neutralizing antibodies, but there are some of them that have been characterized, and as I said, they might get more than 50% of the strains that you would have to test in the laboratory. So we call those types of antibodies BNAbs, for broadly neutralizing antibodies. So this summarizes some work that's been done by a number of different laboratories, but it's been known for at least 15 years that if you do manage to get a broadly neutralizing antibody from these rare patients here, in animal models of HIV infection, they're protective. So clearly, if we were able to elicit these BNAbs, we would have a vaccine. But no one has yet figured out how to make any kind of immunogen that when you inject it, you raise broadly neutralizing antibodies. You can inject things that raise strain-specific antibodies, but as I said, that's not going to do people any good. Okay, so a number of people have decided that, well, right now we don't know yet how to make this vaccine, although a lot of people are working on this, and there's been a lot of progress recently. But you could also think about giving broadly neutralizing antibodies passively, which has been done, for treatment of, let's say, respiratory syncytial virus. That's treated in infants with a purified antibody against that virus, RSV. So people are thinking about giving these HIV BNAbs for passive delivery-based prophylaxis or maybe therapy in HIV-infected people. Okay, so what I want to talk about is a number of ways that HIV evades antibodies and what we might do about it. And I'm going to start with talking about one of the ways that HIV is very good at evading the immune system, and that's through its glycan shield. So here's the virus over here, and it has these envelope spikes here in white. And then if you do an enlarged view of it, here's the spike trimer. The membrane of the virus would be down here. This is a low-resolution view from electron tomography done by Sriram Subramaniam's lab, where they imaged this virus here, and then they took out the individual spikes and did subtomogram averaging. So for a long time, this was the view of, say, from 2008 until 2013. This was pretty much the view people had of the spike trimer. It's pretty low resolution, but it illustrates what I want to say, which is 2 points. First of all, there's a lot of different BNAbs now that have been isolated by many laboratories, the Vaccine Research Center at the NIH, Scripps, Michelle Nussenswag's laboratory, a lot of different laboratories have been isolating these. And people have mapped out where they bind on the spike trimer. So I'll be referring to the known epitopes for these antibodies. But the amazing thing is the antibodies reach in through this extensive glycan shield. This is modeled here. This is not what it looks like really at all, but this is what you'd find in papers up until recently because people didn't really know what the glycan shield looked like. But it was clear that the huge number of glycans on the envelope were blocking access of antibodies, and then the antibodies learned ways to actually interact with those glycans. So the first part of the talk, I want to talk about the complexity of the glycan shield. Okay, so these envelope trimers on HIV are very heavily glycosylated. 50% of the mass of the protein and 30 potential end-link glycosylation sites per monomer. That means around, depending on the strain, that means like about 90 per trimer. So this is a paper from Max Crispin's lab at Oxford, and they did mass spectroscopy on a particular strain of HIV envelope. It's called BG505. This is going to come back because that's the strain we use for structural studies as well. But so they did mass spec, and they identified glycans at each of these potential end-link glycosylation sites, which is asparagine, X, serine, or threonine. Okay, so the point here is that it's heterogeneous. So pink means complex type end-link glycans, and green means high mannose end-link glycans of different types. And then there's intermediate ones. And I'm going to define complex type and high mannose in another slide. But the only reason I want you to see this slide first is to show you that at individual end-link glycosylation sites, say the one attached to asparagine 197, there's both high mannose and complex type end glycans. Now, HIV has a lot of high mannose type glycans for a reason I'm going to describe on the next slide. This is unusual for a protein, let's say a host protein would be mostly complex type. So the way you acquire end-link glycans is that there's a end-link glycosylation sequon here, right here, and then in the endoplasmic reticulum, it gets individual N-acetylglucosamines added, and then it becomes a high mannose structure here. And then that gets trimmed by certain enzymes in the ER to a smaller high mannose structure, and then when it goes on to the Golgi, it can become a hybrid type, which I've removed here for clarity, or it can become a complex type end glycan. Now, the high mannose, there's a lot more high mannose than usual on the HIV envelope, and it's thought that it's because the potential end-link glycosylation sites are so close together that the machinery in the ER and the Golgi doesn't have access to it, so it leaves them at some stage here. And it can't access it enough to go on to make them the more typical mammalian type of glycan, which is complex type. So that's distinguished from high mannose by a core fucose right here, and then a negatively charged set of sialic acid residues here. So complex type N glycans are negatively charged, and then the high mannose are neutral. Okay, so actually, when we do crystallography as someone I've only worked on glycoproteins for my whole career, and we hate glycosylation. We really hate it because it's heterogeneous, and it usually impedes crystallization because it's a chemical mess. Glycoproteins are not really fun to try to crystallize. So what we tend to do if it has a lot of end-link glycosylation is we either treat mammalian cells with chiffonensine, which keeps it here at this high mannose stage, or we express the protein in GNTI cells, which has a deletion of an enzyme that would go on to process it to complex type. So that ends up giving you high mannose only type of glycosylation. And so all of the crystal structures of any type of HIV envelope, be it GP120 monomers, which were a lot of structures have come out since 1998, the first one, of GP120 monomers, they're all produced as high mannose only proteins. And then now more recently, trimer structures have come out. All of them have been high mannose only. And so I just want to emphasize this where we compiled the structures of envelope trimers. We're not going to list all the GP120 monomers, but the first trimer structure, envelope trimer, came out from Ian Wilson's lab in 2013, and then Peter Kwong's lab did a higher resolution version of it, and then various other things happened. And all of these were produced in either GNTI cells or in cells treated with kifenensine, as I told you. And then furthermore, each of these was treated with an enzyme, so it would be complexed with antibodies, and then treated with an enzyme, NOH, which cleaves everything off except the first glycnac. So what you end up is a very small stub of this glycan there in any site that's accessible to this enzyme. This is our structure here from Louisa Scharf that was published last year. We didn't treat with NOH, but it was a high mannose only structure, and we didn't see a lot of ordered glycans. And it's unclear exactly why, but I can come back to that later. But what I want to tell you about is, first I need to point out that about in May, Peter Kwong's lab also didn't treat with NOH, and they got 3 structures of individual envelopes from different clades, and these were described as fully glycosylated. So they are fully glycosylated, but they're high mannose only. And so they saw a lot of glycan, compared to these that were treated with NOH, they saw a lot of glycans on those structures, but they're high mannose only. So they're missing, for example, the negative charges that you'd see in the complex type glycans. Okay, so then this is the structure that we just published. Harry Gristic was the first author, postdoc in my lab. And for some reason, which I still don't understand, well, first of all, Harry decided that he was not going to use these mutant cell lines or treat with a drug. He was just going to try to crystallize the natively glycosylated, totally chemically heterogeneous envelope trimer that we produce. If I'd known he was doing this, I would, of course, told him not to because it wasn't going to work. But he didn't tell me he was trying this. And for reasons that I would say we still don't quite understand, he was able to get 2 independent structures at reasonably high resolution. These are not great structures in general because it's hard to get these to crystallize at all, but 3.5 angstroms, 3.9 angstroms. And this is fully glycosylated, and it's natively glycosylated. So we have a mixture of all of these complex type glycans with the negative charges. We have the high mannose. We have whatever's there from the protein that's produced. So this is an overview of the structure. BG505 means the envelope trimer that we used. It's a soluble native-like envelope trimer that is called a SOSIP by John Moore and Rohir Sanders, who developed this type of soluble envelope trimer. And then the glycans here are in cyan, but there's 2 antibody, the Fab regions of 2 antibodies here. One is 10-1074. Both of these are isolated from Michelle Nussenswag's lab, our collaborator at Rockefeller. So 1074 is in clinical trials right now in humans, in trials that Michelle is directing. And then a new antibody called IOMA that binds to the CD4 binding site. And I'm going to talk about both of these. This was isolated by Lotte von Boehmer in Michelle's lab. Okay, so, and then I'm going to be showing you the trimer with the 3-fold axis vertical, and then we rotate it to look down the 3-fold axis. So I'll usually show those 2 views. Okay, so one of the things Harry did was that when we purify this is the envelope trimer, BG505 is the strain, GP140 is another name for the type of construct we're using, expressed in wild type cells. They're not going to do anything strange to the glycans. Like everything, we run over size exclusion chromatography, and it runs as a reasonable peak. But what Harry did was he separated the fractions so that he used a blue set of fractions here to set up crystallization trials with these antibodies. And then he used this green set. So the blue set migrates higher on the gel. It's higher glycosylation, and then the apparently smaller molecular weights one have less glycosylation. And from the blue samples, I'm just showing you the trimer now, he got the 3.9 angstrom structure. And from the green samples, he got the 3.5 angstrom structure. And you can see pink is complex type N glycans, cyan is high mannose. You can see that there's This is less heavily glycosylated, and it diffracts to higher resolution as well. Okay, so this is a comparison between the envelope trimer structures that are published now. So this is Harry's structure right here that I just showed you with these two antibodies bound. This is a cryo-EM structure solved by single particle cryo-EM by Andrew Ward's lab to 4.2 angstroms resolution. Of course, there's no problem with including all the glycans in single particle cryo-EM because you're not trying to crystallize it. So they were able to get quite a reasonable resolution by single particle EM, and they were able to build in some complex type glycans, but not a lot of the glycans were ordered up on the top of the trimer, so they just built them as high manos. So they didn't see a lot of ordered end-linked glycans, which can sometimes happen. So between particles in their EM single particle structure determination, there was enough heterogeneity that they can't really see a lot of end-linked glycans. This is the highest resolution envelope trimer that's been solved so far from Ian Wilson's lab. It's at 3 angstroms, and it's been severely truncated in its glycans. And this is a fully glycosylated structure of the same strain that we're working on from Peter Kwong's laboratory, and it's fully glycosylated, but it's high mannose only, so it's missing the details of the complex type and glycans. So this is a movie of the structure. The 1074 fabs are in this greenish color, so there's 3 of them because it's a trimer. The Ioma fabs the 3 of them are blue. The envelope is in light brown and light pink for GP120 and for GP41, and it's really not black. Okay, so then the complex glycans are pink, and then the high mannose glycans are cyan. And these red parts right here are something that I'm going to come back to at the end of this section of the talk. Because we've got coordinates for the glycans, we can do surface accessibility calculations and figure out what surface area is free, not covered by glycans. And so there's this huge, well, it's huge for this protein, but there's a set of, there's a place right here that we predict would be targeted by antibodies. I'll come back to that in a moment. Okay, I just wanted to say for the crystallographers in the audience that building these end-linked glycans was not easy because there isn't a precedent for something that's this heavily glycosylated in the protein data bank. And the density, because this is chemically heterogeneous, and it's structurally heterogeneous as well. So the density was not easy for Harry to build, but at each end-linked glycosylation site, this is just an example of 2 of them. He built into 19 end-linked glycosylation sites, and in the supplemental information, he shows electron density maps for all 19 sites. I'm only showing you 2 of them at these 2 glycosylation sites. So he has 2 independent views of this because he has the 3.9 angstrom map and the 3.5 angstrom map, which were solved and refined totally independently of each other. And then what he has is he does conventional difference Fourier maps, and then for those who understand this, this is a annealed omit map. So it's a composite annealed omit map, so there's no model bias at all in this type of map here and here. So then he builds this, and then he assigns it in these schematic diagrams right here. So you can see that at this site, he interpreted the density differently at the 3.9 angstrom structure and the 3.5 angstrom structure. So this is tetraantenary, he built it. This is not. And then, but in each of them, he sees this core fucose right here, which marks it as a complex type glycan. And then he sees sialic acids on the pink ones on the 3.5 angstrom. So that's some negative charges there. Okay, and then here's what he interprets for this site. And he compared it then to the highest resolution structure. No, sorry, this is the cryo-EM structure, which is natively glycosylated, but they couldn't see the glycans at these positions. They were disordered. And then this is the fully glycosylated high mannose only structure from Peter Kwong's lab. So at this particular position, and this is the mass spec assignment. So for all of these, we've gone through and done these comparisons. But without going into a lot of the chemical details, I just wanted to say that we now know the complete epitope of this broadly neutralizing antibody in the context of native glycosylation. This is important because this antibody actually reaches through to contact an underlying peptide down here. It has a sequence GDIR. This has been described extensively by Ian Wilson in the Scripps group. So these types of antibodies reach through this massive glycosylation to contact conserved residues here, but then they're flanked by all of these glycans. And so in the previous structures of these types of antibodies, they were all high-mannose only glycans. So that doesn't matter for the main glycan that it recognizes, which is asparagine 332. This is known from a lot of mass spec and other data to be high-mannose only. So this site is relatively homogeneous. The maps were beautiful in this area, and it's a high-mannose M9, mannose 9 glycan. But these two glycans here that flank the site were actually, one of these was built as a complex type glycan, asparagine 156 by Harry, because he could see the core fucose. And so that makes a difference because although in this structure here we couldn't find the sialic acids, they were disordered, they were ordered in the 3.5 angstrom structure. He could actually see them. And so that means that there's kind of a cloud of negative charge here. We're probably not seeing all the sialic acids. Some of them must be disordered. So there's a cloud of negative charge here, and what does that mean for recognition and raising antibodies? So we've been thinking about this for a while in terms of what does it take to actually raise a broadly neutralizing antibody? And so one way of trying to address this is to solve crystal structures of the germline, that is non-matured form of the antibody, and compare that to a structure of the mature form. So hopefully you all know that when antibodies, so you stimulate a B cell that has a B cell receptor, a membrane-bound antibody that is specific for your antigen, and then in processes of somatic hypermutation, different antibodies get produced, and the ones that bind better are the ones that go out, and eventually the B cell goes in the periphery and secretes a soluble form of that antibody. So that's the mature form, and it has many different hypermutations. It can have a lot, as I'll show you later, or it can have only a few. But anyway, this one, the mature form, is the one that's isolated from the patients, and then you infer what the germline gene is. So Louisa Scharf in this paper earlier this year solved structures of mature and germline forms. And so what you're seeing here is an electrostatic surface where red is negative and blue is positive. And then we've tried, this is of the antibody only. So then we've mapped onto the antibody approximately where the glycans would contact it. So you can imagine this is sort of where the antigen would bind on this surface right here. And then this is approximately where the glycan would fit. And so you can see that for the N332 glycan, there's not a big change in, it remains mostly neutral. And that glycan, remember, is high-mannose only. So that's known. So you don't expect that to change. But the interesting thing, at least to us, is that in the footprint, this is now a different view of the same antibody. You've just turned it 90 degrees of the combining site. Where the 156 glycan would bind, it starts off as not very positive, but then it becomes very electropositive, especially down here. And remember, we don't know quite where probably the glycans are flexible. So we're thinking that as a general thing, what somatic hypermutation has to do in making antibodies against this very heavily glycosylated protein is to become more electropositive because the complex type glycans are electronegative. So we're thinking that that's one thing that needs to be thought about when you think about how to raise these antibodies in vaccine trials and so on, is that the antibody needs to somehow accommodate the fact that the antigen it's recognizing has a cloud of negatively charged sialic acids on it in certain places. Okay, so I then want to switch to talking about the other antibody that was in this crystal structure. So that antibody was called IOMA by Lotte von Boehmer, and it's a member of the class of antibodies that binds to the CD4 binding site. So CD4 is the receptor. If an antibody binds there instead of CD4, it blocks the virus from entering. This is from a review article by Peter Kwong, this figure here. Actually, it's a paper that solved a lot of different structures of CD4 binding site antibodies bound to GP120s. And so there's a class of CD4 binding site antibodies that mimic the CD4 structurally. So the heavy chain down here kind of mimics what CD4 looks like, and then they bind and block the site. And they all come from a particular IgG VH gene, immunoglobulin VH gene, that's called VH1-2. So, and they've been isolated from like 9 or 11 different donors, and they all come from this VH1-2 germline gene. Now, this has been the topic, this has been a goal for a number of labs to try Immunogen design. So basically, Bill Schieff's lab has made immunogens to bind to the germline B cell receptors of these VH1-2 derived antibodies, because the normal antigen won't bind to the germline receptor. So he's engineered these engineered outer domains of GP120 that do bind. And then Leo Stalmatatos has made an intact GP120 that also binds to a germline receptor. So this is, hopefully, people are trying to use these to elicit this class of broadly neutralizing antibody. And there's been a lot of progress in this area recently, but nobody's there yet. Okay, so these are immunogens that are designed to bind to this class of antibody that's only from the VH1-2 gene. Okay, so then LADA isolates IOMA, and IOMA is a VH1-2 antibody also. But it's different from all of these because it's an absolute requirement for these types of antibodies to have a complementarity determining region on the light chain. The third one, the CDR-L3, has to be 5 amino acids long, or it doesn't fit. And only 1% of light chains in the human repertoire, or the mouse repertoire for that matter, have this very short CDR-L3. It's really unusual. So it's going to be hard to raise these antibodies because this is such a rare thing. But the great thing about IOMA is it's got a more normal length. It's right here, 8. And so we think it might be more easily elicited. Now there's another thing about IOMA that's good as well. So this is a coverage curve where you plot the percent of viral strains of HIV that are neutralized as a function of concentration. So the better antibodies go up to 100%. Well, none of them actually do, but that would be ideal. And then they're over to the left if they're really potent. So here is the parental type of VH1-2 antibody called VRC01. So it's this green dotted curve. So it's quite good, goes up to about 80 or 90% of strains, and it's quite potent. IOMA is not very good compared to VRC01. It's this red line here, but it's not as heavily somatically hypermutated. So for VRC01, it's got 41 somatic hypermutations in the heavy chain at the amino acid level, 25 in the light chain. But IOMA only has 22 and 15. So it's going to be very hard to elicit an antibody with that much somatic hypermutation. To give you, normal antibodies are, not against HIV, or more like 9, 12 somatic hypermutations. So this isn't bad. It's a little bit more than normal, but it's certainly not as hypermutated as this other class that people are trying to raise. So we're suggesting that this might be a really good vaccine target. And so fortunately, we know about how it binds now from Harry's structure. So here's the IOMA Fab. You're seeing 2 of them because remember this is a trimer. And it's flanked on either side by carbohydrates. And actually the one attached to asparagine 197 is a tetraantennary complex type glycans. We can see the sialic acid. So again, there's going to be negative charge here. And then the 276, that's the 276 glycan as well. But they're both complex type and glycans. Okay, so then I wanted to just say quickly that because we know now we know more about the actual geometry of the native glycosylation, the native glycan shield, at least from this one example, we can do calculations to plot accessible surface area. And so what's being done here is to show the trimer only. The antibodies have been removed. So threefold axis is vertical here, and it's vertical right here too, and then you're looking down on the threefold axis in these two views. Okay, so these are the side views and the top views. Okay, so areas that have contiguous accessible surface area are colored in red right here. And so these, now you can see them better. I'm showing you two sites here, but remember this is a trimer, so it's threefold symmetric. So there's really only one site here, okay? And then that's accessible surface area plotted, but what's plotted on this view of the trimer is sequence conservation, with low sequence conservation among the strains in white and higher in dark purple. And you'll notice that this same area right here has low sequence conformation, and it's got accessible, solvent accessible areas. And so we think this contiguous area here would be a site that would be potentially accessible to antibodies, but it's not a good site because it's got low sequence conservation. So you're gonna, if you use this as an immunogen, which is being done right now, you're going to get strain-specific antibodies, and that's actually the topic of this paper here by Laura McCoy from Dennis Burton's lab. It's called Holes in the Glycan Shield. They're a target of trimer-elicited neutralizing antibodies. It was just published a month or two ago. It's exactly this site. When they inject this into rabbits, they get neutralizing antibodies, but they're against this site, so they only neutralize BG505. So more needs to be done to plug up these holes. And then I just wanted to say quickly while we're on this topic that, so N156 and N160, we know what those look like, at least in the crystal structure. So Hao-Ching Wang in my lab has been doing single particle EM to look at structures of some of Michelle Nussenswag's new neutralizing antibodies. And this is one we're interested in called BG1. This is just added, this other antibody was added to stabilize the complex. But it binds to a place that is flanked on both sides by the N156 and N160 glycans. So this structure's now at about 6 angstroms. We're trying to get it to higher resolution, but we're looking forward to seeing how this antibody interacts with those glycans as well. Okay, so I wanted to actually then talk about another topic on how HIV evades antibodies and talk about what we might be able to do about that. So what I talked about so far was the glycan shield. Now another way HIV evades antibodies is by rapid mutation, which error-prone reverse transcriptase, and then so the whole thing keeps changing its sequence, and it changes which glycosylation sites are used as well. But then it hides the conserved regions that it has in interfaces. So these are things that the virus does. So when we started working on HIV about 10 years ago, so I read all the reviews, and they gave these as reasons for why HIV is such a problem. And it seemed like there must be something else, because other viruses have these same tricks. Like influenza virus has a glycan shield on its envelope proteins. It undergoes mutations, and it hides conserved regions and makes them inaccessible to antibodies. So we started thinking that you may have noticed how strange this virus looks. It's got a very low density of envelope spikes. It looks really strange. So when this came out from Kenneth Rue's lab in 2006, this electron tomography of HIV variants, and then Sriram Subramaniam's lab did it, and it just looked very strange. And so in 2010, Josh Klein, who'd been thinking about this for a long time, he was a graduate student in my lab at the time, we published this paper in PLOS Pathogens to say that HIV might be evading antibody avidity by the fact that it has hardly any spikes. So the argument, which is in more detail in this opinion piece here, would go something like this. Okay, so you have your regular IgG antibody. It has 2 identical Fabs, and the maximum arm reach between them is probably about 15 nanometers. Now you take your typical virus, and we gave more examples in the paper, but I'm showing 2. You have a dense array of envelope spikes. And so the antibodies can bind bivalently by cross-linking between adjacent spikes. And this allows you to have avidity. And avidity allows you to overcome intrinsically low affinity by multiple binding. So if you have an antigen that's tethered to a surface and you have a bivalent antibody binding here, If the affinity is reduced through mutation of this envelope spike, then this Fab has a higher off rate and it will fall off. But if it's still tethered by the other one, it's in close enough proximity that the first one rebinds. And so that's a sort of hand-waving explanation about how avidity effects can allow you to overcome low affinity. And in this case, the first antibody you make is going to be high affinity, but as the virus mutates, the interaction between an individual Fab and the antigen becomes weaker. Okay, so in the case of HIV, it has hardly any spikes, and those spikes are pretty far apart. And as far as we know, they're not able to laterally diffuse on the membrane, at least not on the timescale of antibody binding. Okay, so you've got these things, they're fairly far apart, so most of the time, just one Fab is going to bind. So what does that mean? Well, we think what it means is that when you have antibodies against HIV that are against this spike, which is the only target of neutralizing antibodies on HIV, they're going to be binding, for the most part, monovalently. And so it would look something like this. Now that shouldn't matter, though. If HIV didn't mutate, that would make no difference, because these would still block entry. But the fact that HIV mutates means that the intrinsic affinity here is going to get weaker, and then there's no avidity to compensate for that as the virus mutates. Okay, so we wanted to fix this. So we thought, okay, we're going to engineer antibodies that can cross-link between these spikes. But if you think about it, there's no universal way to do that, because the distances between the spikes varies even on a single virion or a single strain of viruses. There's just no consistent way to do that. So I'm going to call this interspike or between spike cross-linking. So we thought that the way we could solve it instead was to do intraspike, so within a single spike cross-linking. So then you'd have bivalent binding to every single spike. It doesn't matter how far apart they are. None of that matter. It doesn't matter that there's hardly any. They'd all be binding with avidity despite the low spike density. So we want to make something that looks like that. So how do you make these bivalent reagents? We want to make them bind to two of the same epitopes on the trimer, or maybe two different epitopes, or maybe actually we want to make things that bind to three epitopes that are trimers. So how do you do this? Well, you could do it based on what you know from the structural biology of envelope trimers, but it's too complicated, I think. From Subramaniam's work of electron tomography on viruses, it's known that envelopes on the surface of viruses exist in at least 3 different conformations. So in their papers, they defined a closed state. This is a cartoon of the trimer here. For reference, the CD4 binding site is in yellow. And then when they bound another antibody called B12, it kind of opened a little bit. And then when they bound another antibody called, when they bound CD4, the host receptor, it opened completely. And so that's the open state. So what you're looking at is trimers from the side, trimers from the top, and then coordinates fit into those. So you can get an idea that when CD4 binds, the GP120s kind of open up, like the thing is opening up like a flower unfurling right here. Okay, so we don't know what's going on the virus. So what we decided to do was to use a ruler and we used double-stranded DNA as this ruler. So the reason we chose double-stranded DNA was that its persistence length is probably around 500 angstroms or 150 base pairs about. And then it has a ruler increment of 3.4 angstroms per base pair for conventional double-stranded bDNA. So the idea was if we linked Fabs from an antibody with different lengths of double-stranded DNA, then we could measure the distance between adjacent sites. So Rachel Galamidi, in this paper here, described how you can link Fabs covalently to a little piece of single-stranded DNA. You make a linker between them, and so this is atomic force microscopy of what one of these looks like with one particular length. And indeed, the linkers appear to be rigid. And then she did an experiment multiple times in the paper. So the idea was, you should be able to see increased avidity through an increased neutralization potency. So if the linker is just right, then 2 Fabs should bind bivalently to single spikes to get intra-spike cross-linking, so that would show up as an increased potency. If the linker is too short, it should bind monovalently, and if it's too long, it should bind monovalently. Okay, so then what she did was to measure the neutralization potency for this particular type of, we call it a homodifab because it's the same Fab on either side of the double-stranded DNA. And then this is the neutralization potency here. This is the length of the double-stranded DNA. And you can see that there's a very sharp best distance here that works for the greatest potency. And that was at about 62 base pairs, which is about 210 angstroms. So she did this then with multiple, she did it with this type of homodifab against this strain, and a different type of homodifab that's very similar against this strain. And each time, although the distances they weren't as sharp here, the length dependence, it was always around 60 base pairs. And that actually, if you do the measurements, corresponds to this open structure here. We thought it would be the closed structure that would be most potently neutralized, but it appears to be something is opening up that we can neutralize best if we have the Fabs separated by that distance there. Okay, so are these increased potency compared to the parental IgG? Yes, so this is against many strains. It's like 25 different strains of viruses. This is a coverage curve. And so what you can see is that consistently, the homodifab separated by this right distance, the correct distance, or the best distance, is more potent than the parental IgG. And this is really interesting because, of course, this IgG has 2 Fabs. This has 2 Fabs. But the Fabs are separated by what we think are the optimal distance here, and they're not separated by the optimal distance here. And consistently, it's better. So avidity, we think, this shows that avidity matters. So Rachel went on to make these. This is the result you saw on the last slide. This is a homodifab, same Fab on either side. So now she combined that Fab with another type of Fab that binds up at the top right here, and to make a heterodifab. And that is amazingly better, in fact, than the parentals. So the parents of this are PG16 and 3VNC60. These are the IgGs, and that's how potent they are. And this is the heterodifab, goes up to 100%, and it's just dramatically more potent. It's a 100-fold mean, it's over a 100-fold geometric mean increase in potency for this reagent across all of these strains. Okay, so I always get asked, like, what's the use of this? I mean, first of all, I think that it's really useful because it's much more potent. So if you were going to use it for passive delivery, you could use it at lower concentration. But what I really want to do is stop HIV from effectively escaping. And I think this could be done through avidity. And this gives you an idea of preliminary data that suggests this might actually work. Okay, so what Rachel did was to do in vitro evolution studies where you take cells, you infect them with a HIV with a fluorescent protein reporter, and then you have a certain number of infected cells in your population. And then you treat them after they're infected with either the parental IgGs or a non-covalent mixture of the parental IgGs with no DNA attached to them, or you treat them with the heterodifab. Okay, so what you can see is that you start off, you have 10% of the cells infected, and when you add the antibodies, that goes down because you're neutralizing HIV and not allowing it to infect other cells. But then in the case of the 2 parental IgGs and the non-covalent mixture, that comes back up as the virus mutates to escape. But at least in as long as we were able to get the experiment to go before the cells got contaminated, it looks like we're going down, where there's a downward trend here. So we're very excited about this preliminary data suggesting that, in fact, avidity can be used to suppress successful viral escape routes. So this is now a summary of this. We're interested in this technology because you get dynamic information about envelope conformations, and we think that, like, different types of viruses, some are easy to neutralize, some are hard to neutralize. We think that they'll show different types of length dependencies, so we're comparing viruses with this method. We got really large increases in geometric mean potency. And that's exciting because it supports this hypothesis that the low spike density is one of the ways HIV has evolved to make itself evade antibodies. And we think also the ideal therapeutic then would be something that utilizes avidity to achieve intra-spike cross-linking. We think this should be done with protein-based linkers, not DNA, for various immunological reasons that I can discuss. But if you could do this, HIV's low spike density would be irrelevant. We hope it would be resistant to envelope mutations based on the in vitro evolution results. And then if we have things that recognize different epitopes, that's yet another way to hopefully get around envelope mutations. Okay, I just want to, I'm running out of time here, and I just want to show you some very recent results. We're really interested in this open state right here, and we've been trying to crystallize it for a long time. And this paper I alluded to from last year from Louisa Scharf, we were trying to get a CD4 bound structure. So she put this antibody in, and she put CD4 in. She purified it on a sizing column. All 3 proteins were there. CD4 was there. She solved the structure, and CD4 was gone. And she did it twice, and CD4 was gone both times. Can't get this to work. Kicks it off. But Hao-Ching Wang got a single particle EM negative stain structure with CD4 bound. That was last year. And then this year, I think the paper came out yesterday in PNAS Online, he got an 8.9 angstrom single particle EM structure with CD4 bound. And it reveals how HIV opens in order to bind, after it binds CD4. So this is the structure of the closed trimer, and it's got V1, V2 loops at the top, and they hide the blue part, which is the co-receptor binding site. They hide it so that the co-receptor won't bind unless CD4 first binds. Now, when CD4 comes in, which now it binds here, V1, V2 moves by over 40 angstroms. So the final structure that Hao-Ching solved is the end result here. The rest is an animation. And CD4 actually contacts, so this is just a monomer now, and you're seeing where the V1, V2 loops are. And then when CD4 comes in, you'll see that the V1, V2 loop moves by this 40 angstrom distance, and then it contacts the bound CD4. So that's what stabilizes V1, V2 in this new conformation. And then that allows the co-receptor to bind. We can't see the co-receptor binding loop. It's disordered in this structure. Okay, so I want to stop before the end of the hour. So I'm acknowledging people who did the work. So the natively glycosylated envelope structure was Harry Gristick's structure. He's a postdoc in the lab. Lotta von Boehmer from Michelle Nussenswag's lab at Rockefeller isolated the IOMA antibody. Anthony West did many of the calculations. The single particle EM I showed was a graduate student named Hao-Ching Wang. And then the Interspike cross-linking reagent project was Rachel Galamides. She recently got her Ph.D. And then Anthony helped with that. And the hypothesis was Josh Klein's. Okay, thank you.